Welcome to Slice 7. So, in the previous vids, we've covered quite a bit of ground. The very last one, we talked about, where have I got it? Oh, here we are. We talked about platelet tethering, which led to activation, and then eventually ended up in inside-out signaling. Now what we're going to be talking about is what happens to activated platelets, and that's what we're looking at on the screen. So I think the best way to do this is to kind of step by step break it down. So if you remember what our platelets looked like originally, we called them, actually let me do like this, sorry, let's get rid of that. We'll move that up out of the way a minute. So we called them discoid shapes. So they start off as a disc. So once the platelet becomes active, things start to change. So the platelets go from being a disc via myosin contraction to being a sphere. So we've got this kind of shape like that. Okay, so that's myosin contraction. Now, the next thing that happens after that is that the shape will start to change again. Okay, why? Well, the whole point of this, if you remember right back, is that we've got a damage here in the vessel wall. We've got our von Willebrand, which is adhered to it, and then we've got our platelets. And the idea behind all of this is that we need to get an interaction between the platelet and the vessel wall. Okay, so whenever we've got a situation where we want to get a close interaction, we need to make sure that the thing that we're trying to focus on is going to be a shape that is going to allow it to have as close a possible interaction as we can arrange. So the first thing is going to be this myosin activation, and that's going to change the platelet from being a disc into a sphere. The next thing that's going to happen is all of this platelety goodness, okay, which is a true change in shape. Now this, let me move this over here and just delete this. So this is all going to be about not myosin, but actin. I think we mentioned this when we were kind of touching up on the previous slice, and we said that it was the activation of the platelet was going to start a conformational change in its actin cytoskeleton. Well, now this is what we're looking at. So we've done the myosin changing from a sphere, from a disc to a sphere, and now the actin is going to start to take over. And it's going to start to the actin cytoskeleton itself is going to become modified, and it's going to start to bring out these things here. These things. Okay, so these these things are called fill podia. So what are they? They are extensions. Okay. So what kind of happens, if you think about it, is this platelet squeezes itself into a sphere, and then it pushes out these actin filopodia, that I'm kind of outlining now, and then in behind it, the platelet then kind of continues to change shape. That then starts to fill in the gaps, and you can see it if we're just looking at this area here. Okay, so look at the difference between these two. You can see that the gaps between these are getting filled in. You can see it there versus here. Okay, so these are what's called, so we'll call this stage 1. Stage 2 is going to be these larger, what's called flatted, and these are called lamellipodia. Lamellipodia. And what do? They kind of fill in the gaps between the filopodia. So now we've gone from a disc to a sphere to projections out of that sphere, okay, to filling in the gaps in between these projections. And what you can see, if you examine stage by stage to this, is that as that's happening, all of the granules in the platelet are getting pushed to the center. So it's almost like that as this filopodia is working its way out, it's at the same time pushing the granules towards the middle of the platelet. So we can say that we squeezes granules to middle. Okay, so that's the first bit. And the first bit is the sort of what is happening. The next bit, I suppose, is more, well, why is it happening? So if we just if we come back to our diagram here, we've got this platelet that is nicely tethered, and I suppose we could do like this, I suppose. Put these two side by side like this, and then we can say, right, what's causing it? And what's causing it is drawing an orange. Yes. So we know that orange is platelet glycoprotein 2b3a, and platelet glycoprotein 2b3a is responsible for binding platelet to platelet using a different molecule. And that molecule is fibrinogen. So fibrinogen is going to be the thing that binds platelet glycoprotein 2b3a together. And what is its job? Binds platelet to platelet. So that's kind of really the main take-home point of this, is that we get, after we tether these platelets and they become active, they start to change their shape. Okay, so the change in shape is disc to a sphere, point 1, then point 2, we get activation of actin, filopodia, extensions, and then larger flattened lamellipodia kind of filling in the gap in the middle. What does that do? That squeezes all of the platelet granules to the middle of the platelet, and what causes that? That is this, the binding of platelets to platelets. Okay, so at the same time as we get the platelet tethering itself onto the von Willebrand factor, what we also get is this activation of platelet glycoprotein 2b3a, and then via fibrinogen it binding itself to other platelets. Right. What happens now? Okay, well, we've got this thing here, the increase in cytosolic calcium concentration. And what have we got? We got platelets that have now started to move their granules to the center of themselves. Okay, so remember, we've got this increase in calcium, and what this calcium is going to do is going to cause the alpha granules alpha grans and the dense granules to start to become released. What's in those? Everything that we possibly need, okay, to cause a clot to happen. Chiefly, what you will find in these alpha and dense granules is more fibrinogen. The other thing that we're going to find is coagulation factor 5, which is going to become very important later. This one in particular is found in the alpha and dense granules in concentration 30 times what you would find in plasma. And you're going to see over the next few slices exactly how important that is as well. Now, the other thing that's going to happen, or going to be released, is serotonin. Okay? Now, why do we have that? Serotonin has got two functions. One, it's a vasoconstrictor. And two, it also activates more platelets. So how's that for a bit of inside-out signaling? The other thing is that's going to happen is that the platelets along their outside, they're going to start to reorganize all of their platelet glycoproteins as well. So if you imagine the platelets being like this blue tack with Lego pieces stuck on it. They're going to start to reorganize all of that to make sure that all of the glycoproteins are where they need to be. And then something really interesting is going to happen, and that is that the platelets are going to start interacting with each other. And what we get if I can actually get this pasted on, is this. So what we're looking at in this diagram here is this is like almost the journey of a platelet. Okay, so what you're seeing on this bit here is you're seeing tethers forming with von Willebrand factor on the platelet actual surface itself. And then what you're seeing over this side is what's called a stable aggregate starting to form. And what you're seeing is platelets starting to tether themselves to other platelets. At this point here, this is called this platelety kind of looking blob over here. That is called aplatelet aggregation. The crucial thing is though that this bit of aggregation here, this is reversible. Okay. Now, at this point, what you can still see is that these platelets, at this point where this is all reversible, are still discoid in shape. Over here, you will notice that you can actually see the granules pushed in towards the middle of the platelet, and you can see that at this point they've changed and they've become fully active. At this point, this is an irreversible aggregate. In order to break this up, we would need to actually literally break the clot up. So basically, a nice easy way of thinking about this is kind of almost like the memes do. And that is that platelets are very sociable cells, and they like to have a party. Now, I think this is a fairly good way of doing this, and I think this is a fairly good way of thinking about it. So this is my last little bit on platelets, and what I want to leave you with is kind of this idea of these. What we've had is a series of events, and these series of events have been mediated by chiefly two things. We've had von Willebrand factor and collagen. Now, these mediated events have resulted in platelet adhesion. And the platelet adhesion and the subsequent activation has given us a platform. What is that platform for? Recruiting more platelets. So what is that? Aggregation. Now, we've got ourselves to kind of the bottom two of this diagram, where we're either in a reversible or a stable sort of platelet plug. We now move on to the next bit of haemostasis, and that is secondary haemostasis, otherwise just simply known as coagulation.